Let's break mitosis down into four main phases. As I mentioned, interphase, which happens before, and cytokinesis, which happens after, are not technically part of mitosis, but they are part of cell division. For that middle part, the phases of mitosis are prophase, metaphase, anaphase, and telophase. When it's time for the quiz and you're trying to remember the phases of mitosis, just remember PMAT. Please make alligator tacos. You could probably come up with a better mnemonic than that. There's plenty of good mnemonics to help remember. Interphase technically occurs before mitosis. This isn't included under the umbrella of mitosis, but we're going to lump it in here so we can talk about the process of cell division from start to finish. Remember that interphase takes up 90% of the time of cell division because it's when all of the complicated DNA replication is happening. So in interphase, DNA hasn't been replicated yet. The DNA is still in loose strands. It hasn't been coiled by packing proteins into chromatin in chromosomes, but now there's twice as much of it. And because it's not packed up yet, chromosomes are not yet visible. So at this point, this brief snapshot, you could consider the cell to be tetraploid, having 4 copies of each chromosome. The parent cell already has 2 copies of each, and now each of those is duplicated. That way each daughter cell is going to end up with a pair of each chromosome. This is why condensed chromosomes look like X's instead of straight rods. They're 2 rod-like chromatids joined in the middle. In prophase, the chromosomes will condense and thicken, so all of that genetic material is going to be bundled up into discrete chromosomes. Each duplicated chromosome will appear as 2 identical sister chromatids, the 2 halves of each of those X-looking things. This is also when the mitotic spindle begins to form. The mitotic spindle, or spindle fibers, are extensions of the cytoskeleton, which we talked about in our cell structure lecture. They are long strands of microtubules that are used for moving organelles around. So it plays a big role here. They start to extend, and eventually they're going to latch on and move the chromosomes around. In metaphase, which can be split into prometaphase and regular metaphase if we're feeling extra technical, but I'm just lumping this all into metaphase. The nuclear envelope breaks down into pieces, which frees up the condensed chromosomes, and then those spindle fibers become attached to the center of each chromosome. So the phospholipid membrane that was the nuclear envelope breaks apart, and then the spindle fibers grab onto the central nodes of each chromosome, and those are called kinetochores. This way, the spindle fibers will be able to move them around the cell. Also during metaphase, once they've attached, the spindle fibers will actually move them so that they're positioned along the center of the cell in a straight line. So they're able to line each chromosome in a single file along the center, along an invisible line called the metaphase plate. Then, in anaphase, the third step, the spindle fibers begin to contract, and this is what pulls the sister chromatids apart. So at the end of anaphase, on each side of the cell, there is one complete set of daughter chromosomes. And in the diagram shown here, they only show 4 chromosomes lined up and 4 chromosomes separating. You have to remember that when this happens in humans, there are 46 chromosomes lining up and being pulled apart. It's easy to show a subset in a model like this so that way the image doesn't get cluttered and you lose sight of what's actually happening, which is why they're only showing 4 here. Then lastly, in telophase and the subsequent cytokinesis, now that the sets of chromosomes are on opposite sides of the cell, a new nuclear envelope begins to form around each of those sets. A new phospholipid membrane gets built around each batch of DNA, And then a cleavage furrow divides the cytoplasm in two. So the outer phospholipid membrane begins to pinch in, form a new membrane in between. And then cytokinesis is the actual act of separating into two separate cells. Here's an actual photograph of cytokinesis in human cells. It's been edited to remove all of the other cells around it, but otherwise it's completely packed in by neighboring cells. And the fluorescing dye has been added to the cells so that we can see all the cell membranes and the cytoskeleton. But this is still a real image that's been taken with a microscope, just augmented by highlighting certain colors or adding certain colors. The cytoskeleton and the plasma membrane are not these actual colors. Just two more things to add about mitosis. The first, in plant cell division, we get an extra step because plant cell morphology is a little different. Plant cells have rigid cell walls made of cellulose. So the same process that we just went over, prophase, metaphase, anaphase, telophase, and then cytokinesis, they're being shown here twice on this slide. Up top are microscope slides of cells from a lily undergoing mitosis, where genetic material has been dyed blue. And then down here, the same process has been diagrammed as occurring in a generalized plant cell. It's a bit easier to see what's going on. As the cell divides in two, it's going to need a new portion of cell wall in between the two daughter cells. As mitosis is taking place, there's a number of transport vesicles that are all carrying components of cell walls, so mainly bits of glucose. And they converge in the middle in what's called a cell plate. These little vesicles, these little bubbles of membrane that have glucose inside of them, will fuse and create a pocket where over time the cells create cellulose out of the component pieces and arrange them properly into a new functioning cell wall. Lastly, I want to talk about cell differentiation. This is what allows daughter cells to become specialized for a specific set of functions. If you have two cells that are dividing and creating two genetically identical daughter cells, then if you are born from one cell that's divided many, many times, how do you now have cells that are very different from one another and able to do different kinds of roles? Skin cells, blood cells, muscle cells, neurons. It's all due to differentiation, which happens later on in the life of a cell. Cells in very young embryos are not noticeably different from one another, but as the embryo develops, many of the cells in it acquire unique properties and highly specialized functions. And the process through which a daughter cell becomes different from its parent cell is called cell differentiation. So your heart muscles, your nerve cells, your lung tissue, your taste buds, these are all examples of really different kinds of cells, but they originally had all the same single parent cell. There are 220 differentiated cell types in adult humans. Stem cells are cells which haven't differentiated yet and have the potential to differentiate into any number of different kinds of cells. By the time a human baby is born, it already has most of the specialized cell types that an adult has. Up until that point, the embryo was mostly stem cells, dividing and subsequently differentiating. But newborns and adults still have stem cells in their bodies. You have stem cells in each kind of tissue of your body. But as you get older, the proportion of stem cells to specialized cells gets lower and lower. Keeping stem cells on hand helps us regenerate tissue more effectively and allows us to heal ourselves and replace the kinds of tissues that are being damaged or lost no matter what they happen to be. And this is why so much research has been put into the medical applications of stem cells. So to summarize, asexual reproduction creates genetically identical daughter cells, and this occurs through mitotic cell division, at least in eukaryotic organisms. So mitosis, that kind of cell division, we can break it down into interphase. 90% of a cell's life is in interphase. Most of the time, it's just doing its regular sort of metabolic process, and towards the end of interphase, all of the DNA inside of it gets doubled. After interphase is mitosis. Mitosis can be broken down into prophase, where the DNA condenses, metaphase, where the chromosomes line up, anaphase, where they're being pulled apart to opposite sides of the cell, with one sister chromatid, so one half of the X, being pulled to each side, and then telophase, where the cell begins to pinch and a new membrane forms for each of those daughter nuclei. Then lastly, the full separation is cytokinesis.